Hello, and thanks for tuning in to my talk. My name is Matthew Harber. I am a professor of health sciences at Taylor University. My talk today will focus on skeletal muscle vascularization with exercise. I hope to highlight some important structural and functional characteristics of the muscle microvasculature and discuss how these aspects change with acute and chronic exercise. So I think it's important to start by highlighting that skeletal muscle has a remarkable capacity to increase blood flow during exercise. This graph here illustrates the comparison of cardiac output and blood flow distribution at rest, which is the small circle here, and at exercise, the larger circles, in an average adult and an elite endurance athlete. The size of the circle denotes total cardiac output, and then the color within the larger circle denotes where the blood flow is being redistributed. So you can see here at rest, cardiac output is roughly 5 liters per minute. About 15% of that goes to the skeletal muscle. Now during exercise, blood flow via cardiac output can increase dramatically, upwards to 20 liters per minute in a normal adult, maybe 30 to 40 liters per minute in an elite athlete. And the vast majority of that additional cardiac output is redistributed to the skeletal muscle. So during peak exercise, 75 to 80% of cardiac output is directed to the skeletal muscle, which could be 20 to 30 liters depending on the quality of the individual. So this highlights that skeletal muscle has an impressive ability to accept blood flow during exercise, which denotes that the vascularization must be quite extensive in order to accommodate that blood flow. So looking at the skeletal muscle vascularization, so capillaries are kind of the end point where the blood vessels interact with the muscle tissue. It's the primary site of nutrient and gas exchange. We'll talk more about capillaries in a little bit. If we move upstream of the capillaries, we get into the arterial tree. So this schematic here denotes a soleus muscle, which is mostly slow twitch or highly oxidative, hence it's noted in pink. The bottom shows a gastroc muscle, which has both slow twitch and fast twitch components. But regardless, we see that each muscle has a feed artery that enters into the muscle. And once inside the muscle, that feed artery will branch off into arterioles, and each arteriole may subsequently branch numerous times, maybe 5 to 10 times, depending on the muscle. And that branching of the arterioles is what perfuses the muscle. So when we look at this arrangement, there are several ways that we can regulate skeletal muscle blood flow. One is to alter the size of the feed artery that brings blood flow globally into the muscle. But then once inside the muscle, changing the size of the arteriole or even the number of arterioles will influence the blood flow within that muscle. Now it's important to note that blood flow inside the muscle is not uniform, and that local factors within the muscle will dictate which aspects receive the biggest or greatest amount of blood flow. And indeed, we see that blend blood flow is directly proportional to the oxygen demand. This figure here shows a compilation of studies on the right here using lower body exercise. That work rate is directly proportional to limb blood flow. And even when we look on the left here using smaller muscle mass like hand grip exercise, we see a linear relationship between the amount of work being performed and the amount of blood flow. And important to note that these studies here are actually in the absence of neurally mediated vasodilation, which has been blocked for experimental purposes. So what these studies show is that there's a strong link between the metabolic demand inside the muscle and blood flow to the muscle, suggesting that there is a mechanism from within the muscle that is primarily driving skeletal muscle blood flow. So when considering muscle blood flow during exercise, it's important to first remember that muscle blood flow is the balance between a systemic vasoconstriction and then local vasodilation. It's very important that the body maintain arterial pressure in order to adequately perfuse the organs, but the skeletal muscle is locally causing vasodilation in order to recruit blood flow to meet its metabolic demands. So a couple of main factors related to regulating blood flow inside the muscle are metabolic factors produced during exercise, adenosine, hydrogen ions, temperature, increase in carbon dioxide. These are all factors that are known to cause vasodilation of the arterioles inside the muscle. Flow-mediated dilation is another factor, whereas the increase in blood flow into the artery and arterioles release nitric oxide, which then causes vasodilation itself. From what I understand, there's neither mechanism is dominant, and in fact, they're likely synergistic. For example, there is some evidence that some of these metabolic factors also cause nitric oxide release. And then there's some nice experimental studies showing that inhibition of one method doesn't completely block blood flow, suggesting that the two mechanisms can account for any deficits within each other. Now, muscle blood flow does not tell the whole story. And so if we look at this eloquent study of heart failure patients. So heart failure patients we know have a reduced exercise capacity that's hallmarked by a reduction in VO2 max. So during cycle exercise or lower leg exercise, we see that leg blood flow, global leg blood flow is not different in heart failure and control subjects. If we then look at the capillary pressure of O2, again, no difference between heart failure patients and control subjects. So similar blood flow, similar pressure of oxygen in the capillaries, but significantly reduced leg VO2 of the heart failure patients. So even though blood flow has been delivered to the muscle, or oxygenated blood flow is being delivered to the muscle's inability to use it is impaired, or the muscle's ability to use it is impaired in the heart failure patients. One reason appears to be related to a reduced diffusive capacity of the oxygen within the skeletal muscle. So the diffusive capacity could be related to capillary or microvasculature inside the muscle. So even though, again, the muscle is receiving blood flow, maybe the muscle is not adequately perfused, which leads to a reduction in the ability of oxygen to be used. So looking a little bit further into the importance of capillaries, so this little schematic here shows the interaction of the capillary bed and the skeletal muscle. So we have blood flow into the muscle, bringing oxygen-rich blood, and then the venous system carrying away deoxygenated blood from the muscle. So when we increase blood flow during exercise, the amount of blood coming into the muscle must equal the amount of blood leaving the muscle. So if nothing changes in the capillary bed, in essence, what we've done is increase the rate at which blood, and more importantly, red blood cells, transverse through the muscle. And indeed, this is what appears to happen. If we look at red blood cell velocity, red blood cell flux, during exercise, the rate is higher than during rest. And so why is this important? Well, if we increase blood flow, the rate, the velocity at which the red blood cells travel through the muscle, we are reducing the transit time, which then decreases the amount of time for gas and nutrient exchange, which then is a detriment to muscle performance. So one way to alter this is to increase the capillary bed, which will then accommodate the increase in blood flow by not reducing transit time throughout the muscle. So let's look a little bit more specific into capillaries. Before we get into some more data, it's important to note that how capillarization is expressed may determine kind of the end result, or may tell a different story. So this is a histogram of skeletal muscle fibers denoted by each circle. and then each little dot surrounding the muscle fiber are capillaries. And so from this analysis, several indexes of muscle capillarization can be made. The first is just simply capillary contacts, which is counting the number of capillaries that touch each muscle fiber. Another common method that capillarization is expressed in is the capillary to fiber ratio. So you can see that each capillary is touching more than one muscle fiber. So it's not a one capillary to one muscle fiber ratio. So denoting the capillary to fiber ratio appears to be a little bit more sensitive than just number of capillary contacts. But then taking into account the size of the muscle fiber is very important for determining capillary density, which is the capillary fiber ratio divided by the cross-sectional area of the muscle fiber. And then also a newer method is the capillary to fiber perimeter exchange index. So these last two methods I'll kind of note as the gold star because they are actual measures of capillary density and appear to be more sensitive measures to muscle function as it relates to capillarization. So that being said, let's look at some characteristics of the muscle that might influence capillarization. So again, another schematic here. This is immunofluorescence image with different fiber types, the green being slow twitch fibers, the white being fast twitch fibers, and then the pink dots here are denoting the capillaries. So we know that fast twitch fibers tend to be more glycolytic, less fatigue resistant, whereas slow twitch or type 1 muscles are more oxidative, more aerobic in nature. And so it's not surprising when we look at capillarization that slow twitch fibers, type 1 fibers, have higher number of capillary contacts, have a higher capillary to fiber ratio, and have a higher capillary density as well. And so this is important because not all muscles are the same. And so capillarization will be somewhat influenced by the fiber type of the muscle. But then also we know that individuals have different levels of fiber type, and that fiber types do play a role in health and disease risk. And so perhaps the fiber type specific differences in capillarization kind of feed into that. Now, if we transition into some age-related differences, we know that age is associated with a reduction in exercise capacity. So not surprising that there's a consensus that not completely unequivocal, but there appears to be a reduction in capillarization with aging, denoted here by reduction in capillary contacts, a reduction in capillary to fiber ratio. Now, in this example, the capillary density itself was not statistically lower in the older adults, even though there's a trend for a reduction, and that's likely due to atrophy of the muscle fibers, so that when you account for the cross-sectional area of the muscle, the fiber, the capillary density is not impaired. But looking at functional aspect, and this is in middle-aged adults, which were, I think, mean age 43 here. This is looking at leg blood flow during exercise. No difference in leg blood flow during exercise in young versus middle-aged adults. But when we look at leg vascular conductance, which is blood flow per unit of pressure, we see that there's a much lower vascular conductance in the middle-aged adults compared to the young. And this is due to a higher blood pressure for any given blood flow. And so important to note here that in this particular study, there were no differences in capillarization. So even though the structural components of the muscle microvascular was similar, the functional components were not. So this denotes that microvascular dysfunction may precede the loss of capillarization with age. So if we transition now into some clinical populations, we know that peak VO2 is a very important marker of health and functional capacity. So looking at peripheral arterial disease patients here, we see that there's a strong relationship between peak VO2 and capillary density. There's also a relationship between peak walking time and capillary density, and then also claudication or the onset of pain during exercise with capillary density. So here we see that even in in clinical populations, that capillary densities appear to be pretty important for regulating functional capacity. Again, the good news, looking again at peripheral arterial disease patients, is that capillaries can be increased with exercise training. And this graph here shows that in the early phase of a at-home exercise training plan, just 3 weeks of exercise increases capillary density in these patients. And then at 12 weeks, there is an increase in VO2 max. But so interesting, the increase in capillary density preceded the increase in VO2 max, which is a theme that we'll see in other studies later in the talk. So that when you look at the relationship between peak VO2 and capillary density, it's significant whether you're pre-trained, post-trained. So again, denoting that capillary density plays an important role in determining peak VO2 in this clinical population. So looking back on the age story, and as we noted previously, aging is associated with loss of skeletal muscle mass, and that may be due to a loss of capillaries. So this particular study looked at older adults with vastly different activity levels. So just looking here is the impact of habitual physical activity, denoted by steps per day, on capillary density and capillary fiber ratio. So again, showing that activity level plays a role in the capillary density with aging. So this particular study had an interesting approach where they split the participants into a low capillary density group, that's the black bars, and a high capillary density group here, that's the white bars. And then they underwent a resistance training protocol. So the low capillary groups actually increased capillarization with resistance training, whereas the high capillary group had no change. So denoting that there might actually be a threshold for which adequate amount of capillary density is necessary for muscle health. And then if we look at changes in muscle mass, the low capillary density group actually did not improve muscle mass with strength training, whereas the high capillary group did improve lean mass with resistance exercise training. So again, this study suggests that having adequate capillarization is necessary for the muscle to respond to exercise training. So to kind of wrap up here, there's actually a very recent meta-analysis here looking at the effects of exercise training on capillary growth. So we'll start first by looking at the influence of exercise intensity. So we'll look at exercise training here, just moderate intensity training, high intensity training, and then sprint interval training. So all forms of all intensities are associated with increases in capillaries, which is good. And surprisingly, low intensity exercise may actually be more advantageous for increasing capillary density compared to the higher exercise intensities, which might be related to the change in muscle fiber size. Looking at initial training response, not surprising that the more fit an individual is, the less likely they are to increase capillaries. So the good news is, looking at untrained individuals, they should be the most responsive to exercise in terms of increasing capillary density. And then looking at duration, as we kind of saw before, capillaries can increase pretty early in an exercise training program. And that's what we see here, is that the initial 4 weeks, there's capillary growth, which kind of persists or levels off with the duration of the exercise training. So this kind of suggests that capillary growth is necessary in the early phases of exercise training in order for the muscle to improve the other aspects of exercise training. So to summarize this systematic review, they concluded that capillary growth occurs in response to exercise training, similarly in healthy and clinical populations, in young and old individuals, and similarly in men and women. So again, kind of take-home message that capillary growth is an important response to exercise training in all individuals. So to kind of wrap up, hopefully I've impressed upon you that skeletal muscle, microvascular structure and function are important in all individuals, that the balance between function and structure needs to be considered to collectively to adequately understand the microvasculature during exercise. We know that capillary density is important for muscle health, and particularly in response to exercise training. And then that we know that exercise training appears to be beneficial for all individuals. So with that, I will end, and thank you again for your attention during my talk.